Hello folks, I want to do a quick video for you on muscle twitches and uh, talk about how muscles, not just individual fibers, contract and generate tension, but how a whole muscle generates tension and the mechanisms it uses to do that. And then say a little bit about the different types of contractions that muscles are capable of. So we're going to do basically these three that are highlighted uh, in green here, in pale green, right? Describing the components, motor units, um, and then explaining these processes that lead to contraction, which are wave summation and recruitment, and then explaining tetanus, and then we'll talk about these different types of contractions, isotonic and isometric contractions, and the two different types of isotonic contractions that we can do. And then um, and then we'll move on to more muscle physiology of generating energy and using energy to generate or to fuel contractions. Okay, so let's jump back over to our lecture, and I want to start here where we left off with looking at the stimulus that creates a single muscle twitch, and we said that that was this thing called an action potential that sweeps across the muscle cell, and it's delivered by a motor neuron to the muscle cell. So I just want us to remind us of this. What we're measuring here when we talk about action potentials is we're measuring a depolarization or a sort of a change in polarity across a little piece of membrane of a cell, in this case of a muscle cell. So we're measuring voltage in millivolts over time, right? So how does it change over time across a little piece of membrane? So you'll remember that this is kind of negative 70 millivolts is like the resting potential of the cell. And then there's this threshold potential at which all of these voltage gated channels, the sodium and potassium voltage gated channels, will open. And so what this action potential is doing is it's a signal from a neuron to a muscle cell that will cause the muscle cell to contract. And so this stimulus that we see is actually the neuron communicating this, transducing the action potential, or sorry, reinitiating the action potential in the muscle cell. And so this initial depolarization as charge floods into the muscle cell is all due to the binding of acetylcholine to those ligand-gated sodium channels at the synaptic, in the synaptic cleft and on the postsynaptic muscle cell. Right, those are the channels that are opening, and sodium is rushing in and depolarizing the cell membrane. Then once we get to threshold, then all the neighboring voltage-gated sodium channels open rapidly, causing lots more sodium to rush into the cell. And as that's happening, the neighboring potassium channels are also opening slowly, right? So there's a very slow increase, sorry, very rapid increase in the permeability of the cell to sodium, and a very slow increase to potassium. In fact, that kind of reaches its peak right here. The sodium influx and potassium channels then take over. So at this point, right, we had the rapidly opening sodium channels. About this peak is when they start to close, right? So you have one and the other, but that's not all at once, but you know, they're all sort of closing right about this same time. And that's right about when potassium channels get fully open. And so we get now repolarization, because remember where we are now is plus 40, way far away from the equilibrium potential of potassium. We saw that was down here at like negative 90 millivolts. And so we get now a rapid efflux of potassium as it leaves the cell, causing this repolarization, right? And then we get this little refractory period as because these potassium channels are slow to open, they're also slow to close. So we get it pushing past the resting potential more towards the equilibrium potential for potassium before we get sodium potassium pump sort of rectifying everything and getting us back to resting state. Okay, so review that if that is still not quite clear. I encourage you to, like we drew it before, to draw it all out so you can see which channels are operating, where those channels are, and which direction charge is flowing through those channels. Now also note the timing of this. We are done in 3 milliseconds. If our stimulus is here, this is sort of the beginning, right? And the action, truly the action potential, right, doesn't start until right here. Because if just a little charge flowed in and you never got to threshold, well, then all those neighboring voltage gated sodium channels would remain closed and you wouldn't get this huge depolarization. It's only if there's enough incoming charge to get this cell to threshold, and then you get all those neighboring sodium channels to open. So really the action potential starts here at threshold. And so we're done by, I mean, we're getting back to resting state in about 3 milliseconds, right? We got a little bit like 1, 2, 3, a little bit more, right? 3 to 4 milliseconds and we're done. That's a really short amount of time. That's going to be important when we talk about contraction later on too, and how contraction can be, how tension in a muscle can be increased or sustained over time. Okay. So now I'm going to show you, this is a totally different figure. So it looks kind of similar, but now we're measuring two different things. And what I'm showing you here is a twitch contraction. So this is like the arrival of a single action potential. And you could think about this happening in a single cell, but really it turns out that a single motor neuron will contact many muscle fibers within a single muscle, a single whole muscle. So this is really sort of looking at tension produced by all those fibers, but it sort of doesn't matter. You can think of it as a single cell. You can think of it as this whole motor unit. You can even, we can even measure tension across a whole muscle cell, which we used to do long ago before we could get to these little individual fibers and piece them apart. Okay. So this is measuring tension here rather than millivolts. So that's like the amount of force produced. This is still measuring time here, and it is in milliseconds, but look how far apart these are. Now we're out to like 40 milliseconds. And I'll tell you, this myogram is for an average muscle. We'll see that muscles differ in how quickly they complete this process of contraction and relaxation. Right? They differ in how quickly they complete this process of contraction and relaxation, but this is on average. It has 3 phases to it, and it's started by an action potential. So there's this latent phase right here, right? You can see again the stimulus. Remember, this is an action potential basically being reinitiated in a muscle cell. So this is after it's already been transduced, and crossed the synaptic cleft, and now it's bound to the sodium channels and already let all that sodium in. So this is the stimulus at time 0. What's going on in this latent period where there's no tension being produced, which means the muscle is not contracting at all, it's still resting, is that action potential is propagating along the sarcolemma, it's down the T-tubules, it's getting to the terminal cisternae, the sarcoplasmic reticulum is releasing its calcium ions and they're diffusing down onto the myofibril. All that's happening in the latent period. So think about all those steps we talked about last time. That's all happening in like 2.5 milliseconds, really fast. Right? So that's the latent period. Then you see this little uptick in tension. That is when cross-bridging begins, right? And so obviously it doesn't happen all at once. You have first a few troponins bound to calcium and moving their tropomyosin and exposing those myosin active sites, and then more and more and more. And as that happens, you get more and more myosin reaching out and grabbing onto actin and pulling, right? And that continues to happen. Remember, as long as there's ATP in there, that pulling continues to happen as long as the calcium is bound, the tropomyosin is moved, and the active sites are available. And as long as there's ATP around, myosin just keeps doing its, cross bridge, power stroke, release, recovery, cross bridge, power stroke, release, recovery, right? They keep doing that. So for about, what is that, 15 milliseconds, right? You get this cross bridging happening and contraction is occurring and tension is building, right? So this is as more and more sites are exposed, more and more myosin is grabbing to actin. That's what builds tension, right? So you can think of this as on this contraction phase, more and more myosin is binding to and pulling on actin. But remember, this is a single action potential. There's one signal, and as soon as all those calcium ions are released, we have those calcium pumps on the sarcoplasmic reticulum shoving them back in. So as soon as that concentration gradient is created and calcium flows down onto the myofibril, it's reversed and they start to go back towards the sarcoplasmic reticulum. So it's not very long that we get this tension, and this tension that's built is not very much until we get to this point where basically you get a reversal, and now you must at this point have calcium ions, more calcium ions leaving than you had on the myofibril. More of those active sites become covered up, and now tension drops as fewer and fewer myosin heads can grab onto actin because they're being covered up again, right, by the tropomyosin. And this is kind of a slower relaxation phase. And all that's done in like 40 milliseconds, right? So this is all in relaxation, calcium reuptake, tropomyosin covering actin. Now there's not as many available for myosin to grab, and we get this relaxation phase. So that's a myogram. So think about this for a minute, and we'll transpose these two, but the stimulus takes about 3 to 4 milliseconds, right? So by the time the stimulus arrives, let's just go 3 to 4 milliseconds later and think about, all right, well in 3 to 4 milliseconds, this cell that's about to undergo this contraction will be reset and at rest once again and ready to receive another signal. This will just be important for the future. In fact, I'm gonna jump back, right? Look at that. There's the signal. 1, 2, 3. By 4 milliseconds, we're back where we were, resting state, and ready to respond again to another signal. That's right about here, right, in this contraction phase. It's like at the very beginning of this contraction phase, it could potentially receive another signal to contract. Okay, so here's that these lines are a little bit thick, but look, here's a skeletal muscle action potential, contraction. Unfortunately, it can't even fit the milliseconds on here, right? But the point is, this is now ready. Here we are at resting state again to signal another muscle, or this muscle cell is ready to receive another signal by the time you're right here in the contraction phase, right? So you could actually get multiple signals throughout this contraction phase. And this is going to be one way that we actually build tension. So this is an important thing to remember about the timing difference here, about how skeletal muscle has basically reset itself. It's back to resting state shortly after, very shortly after, right? Like maybe right here after a contraction begins. So this leads us to talk about how does a whole muscle achieve a contraction. And there's two methods that are used all the time, pretty much, just in differing amounts and depending on the load that the muscle's trying to overcome. This is called recruitment and summation, or motor unit recruitment and summation. Okay, so we know these single muscle fibers contract when a stimulus arrives. We know a single stimulus produces a single twitch, and that myogram we saw is an example of tracing the tension produced, or recording the tension produced by a single twitch. And we know those twitches produce whole muscle tension. So remember how long one twitch takes, at least on average. What did we see on that last myogram about how long does one twitch take? So hopefully you said about 40 milliseconds. So how do these twitches coordinate? They have to coordinate across muscle fibers and across fascicles, right, to get a whole muscle to contract. And they do that by building tension, not just producing their individual little tension blips, but by stacking that tension on top of each other. So they do this in two ways. You can stimulate multiple what are called motor units, and that's called motor unit recruitment. And what does that mean, stimulating multiple motor units? Well, this means stimulating multiple muscle fibers at the same time. So I told you that actually a single motor neuron, which is coming from our spinal cord, connects to multiple muscle fibers, right? That is one motor unit, one motor neuron, and all of the muscle fibers it connects to. That's one motor unit. So if we stimulate multiple motor units, that means we might be stimulating, let's say a motor unit has 5 muscle fibers per neuron. We could stimulate 3 motor units and we'd get 15 muscle fibers firing, right? So that's one way we can do it. We can do multiple motor units, and we can coordinate those in different ways. So they don't have to all be stimulated at once. You could stimulate 3 and then 3 more and then 3 more and alter their stimulation pattern. And that's really what recruitment is kind of building tension by deciding which motor units to fire at the same time and altering their firing over time. The other way is just stimulate a single motor unit more frequently. So this is kind of what we just talked about with summation, right? If you fire multiple action potentials so that as soon as a muscle cell is ready to receive another signal, it gets another signal, you're basically going to be stacking those signals on top of each other. And what happens, of course, okay, well what happens with each signal that arrives, right? What's the end product of a signal reaching a muscle cell? Well, you know that it's the release of calcium and then a contraction that follows that because of the release of calcium. So if we continue to fire to receive signals that release calcium, that means more and more and more and more and more calcium is dumped onto the myofibril, right? And if more calcium is there, that means more of those active sites are going to, more is going to be bound to troponin, so more active sites will be exposed, so more myosin in that myofibril can bind to actin. So that's another way of achieving tension. We just overwhelm the myofibril of a single cell or of a single motor unit with calcium so that as we get exposure of maximum number of active sites. Okay, so I just want to show you that. So here's our motor units, and this is a little visual or a diagram of what I kind of explained. Is a motor unit is one motor neuron and all the muscle fibers that it contacts. So here, these purple or blue or red, whichever one you want to follow, you'll see this is a single motor neuron. This is a cross section of a spinal cord, right? And this will become important in a few weeks when we talk about the nervous system. All our motor neurons are located in this ventral portion of the spinal cord. So this is anterior or ventral, this is dorsal or posterior. So here this motor unit travels off, it branches and contacts all of these purple muscle fibers, right? So this is a motor unit, right? Each one of these is a motor unit, and you can see that reflected here. So we'll see that motor units vary in size kind of according to the function of the muscle. So some muscles that are big prime movers of the body have huge motor units. In other words, a single motor neuron might be contacting 1,000 muscle fibers, and others that are responsible for very fine movements, like those of your eye, for example, or even some in your forearm that control your fingers, the movement of your fingers, have far fewer muscle fibers. So they have much smaller motor units, maybe 5 maybe 5 muscle fibers per motor neuron. Right. So here, just an example, yeah, maybe 4 to 6 fibers per unit, or big muscles of the legs, big prime movers, like 1 to 2,000 fibers, right, per unit. So these are the things we can vary the timing of these. And let me show you. Do I have? Yes, I do. Okay. So let me show you. I just want to introduce that concept of motor units. That's what a motor unit is. Then let's talk about summation, where we're kind of going back and just thinking of, okay, let's talk about a single motor unit. And this is the tension over time. So we're seeing a bunch of myograms here, right? The tension over time when we have a complete relaxation phase. Now, what does it mean to have a complete relaxation phase? How do we get all the way back to completely relaxed? Well, the only way that that could happen is if we get complete or nearly complete reclamation of calcium ions back into the terminal cisternae of the sarcoplasmic reticulum. That's the only way we can get back to complete relaxation. In this example, where we have basically it looks like a stimulus fired as soon as we get back to relaxation, it turns out that it takes even when we get back to complete relaxation, that doesn't mean that all the calcium ions that were released are back in the sarcoplasmic reticulum, right? It just means all those calcium ions that were released are no longer bound to troponin, or no longer bound to enough troponin to expose the active sites enough to allow any measurable tension to be produced by myosin grabbing onto actin. So it doesn't mean that all the calcium's back in there. It's just not enough to measure any kind of tension. So probably there might be still a few bound to troponin. There's probably still quite a few out in the cytoplasm or the sarcoplasm. They haven't, made their way back to the sarcoplasmic reticulum along that reversed concentration gradient, but they're just not enough bound to troponin to allow myosin to do any measurable pulling, which is why this shows you stimulating right at the end of relaxation, which is why you actually get a little bit of a build-in tension here, is because there's always still some left in the cytosol. So maybe you reclaim 100 of the released calcium, or sorry, maybe you release 100 calcium ions, but by the end of relaxation, you really only reclaim maybe 95 of them. But when a new signal arrives, you release another 100, right? And so this is a little bit of a simple-minded exercise, but I just want to show it to you anyway, to think about the way that calcium ions are released from the sarcoplasmic reticulum. So I'm just going to pop over, to our sketch pad real quick. I'll move this stuff out of the way for us, and I just want to show you. And this is important too for thinking about how much calcium is even stored in the sarcoplasmic reticulum. We've said it's a huge storehouse, right? Well, how much are we talking about? Okay, well, okay, so imagine, what is our calcium? Our calcium is a green ion, right? Okay, so imagine I just have, here's my sarcoplasmic reticulum. Wow, there's my sarcoplasmic reticulum, and what have I got in there? What kind of channels do I have on the sarcoplasmic reticulum? Tell me what type of channels I have. What type of gated channels I have on the sarcoplasmic reticulum. Okay, hopefully you said you have mechanically gated calcium channels, right? So here's some mechanically gated calcium channels, right? Mechanically gated calcium channels. And inside, right, this is inside the sarcoplasmic reticulum, I have tons and tons of calcium ions. So all these green dots are calcium ions, right? And how much is tons? An ocean. Think of it that way. I have an ocean of, or if that's too big to wrap our brains around, which is fine, Green Lake, right? Green Lake in Seattle, a Green Lake full of calcium, or if you haven't been there, Lake Union, a Lake Union full of calcium ions, right? So you know that when these channels are open, some calcium ions are going to be released. Well, when we're talking about a single action potential arriving and causing the opening eventually, right, via the excitation contraction coupling, we're talking about a single action potential opening some calcium channels and allowing calcium to escape. How much calcium? Okay, well, if this is a green lake full of calcium in here, maybe we get a puddle, right? A big puddle of it released, okay? So not that much, right? So maybe there are, I don't know, 30,000 calcium ions in here, and we get maybe 100 released. That's probably not quite the size of it, but it's something like that. So every time we release 100 calcium ions, right? These are our calcium ions. And then by the time we get to the end of the relaxation phase, we've reclaimed 95 of them. That leaves 5 just out here, right? Then we release another action potential arrives and we release another 100. Now, instead of 100 calcium ions that we had on the myofibril, now we have 105 calcium ions. So now we can probably get a little more tension because there's going to be a few more active sites exposed, and that means a few more myosin heads can grab on, right? You see where this is going. We're going to reclaim another 95 to get us back to, now we have 10 still out here, right? Now we have a total of 10 still out here. And then we release we have another action potential, and we're at 110. So we get a little bit more tension produced. So that's how you should think about about kind of how much is in here, right? It's really hard to run out of this stuff, right? Out of calcium ions, even if we're stimulating for a very long period of time, it can happen. But we only release a fraction with each action potential. So there's a pretty big storehouse here. We only release a fraction, and we reclaim, by the time we get back to full relaxation, we've reclaimed a large fraction of it, but not all of it. Okay. Okay, let's pop back over here and look at, this is looking at, now we've had our little increase. Now let's look at this. This is when the relaxation phase is incomplete. So this is when we haven't even gotten to complete relaxation again. When we get another action potential fired, right, we get this steady increase in tension because we haven't got full relaxation. We haven't got complete relaxation. And this is just showing that going all the way up to the maximum tension that the muscle can produce, right? And there is, of course, some maximum tension. That would be when all the when enough calcium is bound to troponin that all the active sites are exposed and all the myosin is grabbing onto the actin filaments. That would be the maximum tension the muscle could produce. It can't do any more than that. Right? It doesn't have any more myosin to pull with. And then this is showing this is what happens, and these are all different types of summation, right? So here we are at complete tetanus, right? Maximum tension is tetanus, right? That's when you have the muscle in producing its total tension it can. This is when you have eliminated the relaxation phase. So here you haven't even allowed the relaxation phase to occur, because look how you've squished the arrival of the action potentials. Now they're arriving so frequently that we never even get a relaxation phase at all, right? This cell gets the signal to contract so fast that that calcium ions are just literally being poured into the sarcoplasm before any reclamation can have any effect. Any reclamation of calcium ions can have any effect on the tension, right? So this little blip is allowing a little bit, this is allowing none. So this is one way to build muscle tension. Of course, this does it a lot faster when we just eliminate the relaxation phase than when we allow it to be incomplete. And that's going to produce totally more tension than we can ever get when we allow for a complete relaxation phase. So let's think about if we go back to our sketchbook and just think about this. If this is when we have a complete relaxation phase, right, and we're in TREP, then this would be an incomplete. What did that look like? I don't know. It looked like, yeah, okay, so let's say we release 100 again, but we only reclaim 50. Whoops, gotta line these up. Maybe we only reclaim 50 because it's really an incomplete relaxation phase, rather than the 95 we were able to reclaim when we had a complete relaxation phase. So we reclaim 50, of course that leaves 50, so we add 100 more the next time, and we're at 150. Look how much the tension now has built over when we allowed a complete relaxation phase. And again, we reclaim 50, so we're at 100, and then we add another 100. You can see we just build tension a lot faster when we have the incomplete relaxation phase. And of course, with a relaxation phase eliminated, right, we get no reclamation at all. So it's just plus 100. You haven't taken zero, you haven't taken any calcium back in, or not an appreciable amount, before we get another 100. So there we're at 200, and then I won't even do the minus, I'll just add another 100. And right, this is when we have eliminated the relaxation phase. So these are all examples of summation, right? That summation just differs in whether we allow a complete, incomplete, or don't allow any relaxation phase to occur. And you can see how that's going to really alter the calcium ion concentration on the myofibril. And because of that, that's going to alter the tension that the muscle produces. Okay, pop back over here. Let's talk finally about recruitment. So recruitment just means firing multiple motor units kind of at the same time. So this is showing if we stimulate a nerve a little bit all the way up to a lot, that changes the number of motor neurons that are excited, and then those motor neurons all communicate with a motor unit. So this is showing a progressive state of stimulation of more and more motor neurons. This is showing like a cross section of a nerve, right? So this is like a cross section of a nerve, which each of these gray circles being a cross section of a motor neuron. So if we stimulate more and more of those, we see that peak tension that's produced, gets higher and higher until we're stimulating all the motor neurons. That means all the motor units are stimulated. That means we get maximal tension. Okay, so how this really works in real time is we typically don't just stimulate all the motor units, but we vary their stimulation pattern in order to achieve a particular tension. So now if we look at tension mapped here and time, we're seeing a bunch of myograms again, but we're seeing myograms of individual motor units. And hopefully you can see the myogram of the red, the blue, and then the purple. Tell me, is this motor unit, this red motor unit, experiencing complete relaxation, incomplete relaxation, or an eliminated relaxation phase? Okay, hopefully you said that's a complete relaxation phase. It's going all the way back down to zero tension, that red motor unit, before it's stimulated again, but it's just barely complete, right? So this is the tension each one of these is producing. It's showing each motor unit and the tension it's producing. But look at here, we're getting total tension that's produced by that muscle. So the tension as measured in the tendon. And you can see that it's higher, right? And that's because we're recruiting these motor units. We're recruiting, for example, motor unit 2 before motor unit 1 has reached complete relaxation. And because we're doing that, right, we actually have these periods here where we have tension overlapping, right? There's some overlap of tension produced by motor unit 1 and motor unit 2, right? Barely by motor unit 1 and motor unit 3. You can see there's this narrow area of overlap between the purple and red, but there is a little bit. But that means we can vary these motor units to produce more tension than either one could produce on their own, right? If they're allowed to do a little relaxation, right? And this becomes important. Relaxation is an important phase for a muscle to go through. It can't just be in tetanus for very long because it quickly exhausts its supplies of ATP and of the fuel it needs to contract. And you know that to keep a contraction going, you have to have ATP to bind to myosin to cause it to release from that actin subunit. So this is how motor recruitment can be used in order to increase tension, right, above what any one motor unit could do without going into summation. Okay. So, and it turns out the motor units even vary within a muscle. So typically in the bigger muscles, we have variation in motor units, right? So some motor units are small, some are medium, some are large in terms of the number of muscle fibers that are connected to a motor neuron. And this is just showing, if you have a light load to lift, you might only stimulate small motor units to do that because you only need a little tension. If you have a heavier load, you might stimulate another medium-sized motor units. And there are, you could stimulate larger motor units too. Okay, so to kind of wrap up here, I just want you to appreciate that what ultimately controls tension that we've looked at is calcium ion concentration. The more calcium ions that are in the sarcoplasm, the more that are bound to troponin, the more active sites that are exposed, and the more myosin that can bind to those active sites. And the other thing, and this is the last thing we're going to talk about here, is the amount of overlap between actin and myosin. This is maybe an easier one to think about. This is showing the degree of myofilament overlap here in all these little diagrams, the degree of myofilament overlap, and that is what determines the amount of tension that's produced, or it contributes to determining the amount of tension that's produced. So it's a limiting factor, right? So the more cross bridges you have, obviously the more tension you have. And this is showing, here is, where are we? Here we are. showing the normal range of a muscle, and then a sort of an overstretched muscle and an overcontracted muscle. And this is showing, again, the tension, the percent of the maximum tension that can be produced, where 100 is the total tension that the muscle could ever produce. This is the length, the resting length, right? So this is showing there's this optimal region right in here where you have, in this sarcomere, total overlap between the actin and the myosin on all these, right? So that means if we were to completely load this up with calcium ions and expose all these active sites, all of these myosin heads would be able to grab onto an actin filament. So this is the maximum tension that could be produced even by this little sarcomere. But what happens as you stretch those apart, right? These interdigitated filaments, if you stretch them apart, well, look at this. Now we only have a fraction of the actin overlapping with myosin. Just this bit here is overlapping, right? So even if we dump tons of calcium ions on it and expose all these active sites, there's nothing to grab onto these active sites. These myosin heads, they're grabbing nothing. There's nothing over there to grab onto. So of course the tension you can produce is going to be less because you have less overlap between these two, and you can just carry that out to the end and see like an overstretched muscle with no overlap at all, right? I mean, if you think about when you stretch a muscle, trying to contract it, and what does that feel like? I mean, it doesn't feel like you're producing much tension when you do that. And that's because you're not, because you can't, because these aren't overlapping, right? But the same thing, or another problem happens when you contract the muscle too much. What happens here? We have complete overlap. What's wrong? Well, now you've got these pushing against each other. So now you've got the actin filaments actually pushing against the anchoring protein for the myosin filaments, right? Look at that. It's pushing against it. That causes interference and doesn't allow these myosin units that are pulling on these actin to pull as effectively. They're basically getting resistance from the opposite side, right? Because these are moving in this direction. So that and then it gets really terrible, really bad, or really really bad, really fast, right? When you do this and you actually push this actin filament past here, so now you're getting interference here, and you're getting into the myosin filaments on the other side. Now what's happening? Well, these myosin filaments are pulling actin that direction. These myosin filaments are pulling whatever actin they grab onto that direction. So in this case, you have these myosin heads pulling that way, but these myosin heads here grabbing onto this same actin and pushing it that way, or pulling it that direction. So you actually get myosin heads pushing the actin against each other. So you get really difficult to build any kind of tension. So this is important to just recognize. This sets the limit for the total amount of tension that could be produced, even if this is completely doused with calcium ions, right? You can't get as much tension if you don't have the overlap, or if you have too much overlap, so much that you're pushing against the anchoring proteins or getting into the opposite side of the sarcomere. Okay, so I think that is where we're going to end. Please let me know if you have any questions about this. I think we covered those 4 learning outcomes in this one, so I will see you again in a future video.